Hi, my name is Sohail. Today I am going to discuss the part 2 of prokaryotic transcription. It includes initiation, polycistronic mRNA, RNA polymerase, abortive transcription and prokaryotic promoters. As we know, prokaryotes, they don't have membrane bound nuclei, and the process of transcription, that is synthesis of messenger RNA from DNA, translation, that is synthesis of protein from messenger RNA, and mRNA degradation, that all takes place simultaneously. Prokaryotic transcription often covers more than one gene and produces polycistronic mRNA that specify more than one protein. Suppose this is the DNA molecule, and from this DNA molecule here mRNA molecule is synthesized. That process is called transcription. In this mRNA molecule in prokaryotes, more than one genes are transcribed. As we can see here is structural gene 1, structural gene 2 and structural gene 3. Structural gene 1 is responsible for the synthesis of first polypeptide, it is responsible for the synthesis of second polypeptide chain and third polypeptide chain. So, this type of mRNA is called polycistronic mRNA. Polycistronic mRNA that codes for multiple different proteins products. Generally, polycistronic mRNA is found in prokaryotes, while eukaryotes, they have monocistronic mRNA that only encodes for one protein product per mRNA molecule. With polycistronic mRNA, prokaryotes, they have less flexibility in regulating gene expression. That's because when they produce that mRNA, all of these proteins products on that polycistronic mRNA are going to be produced simultaneously. So, it is not able to pick and choose to express only certain proteins products on that polycistronic mRNA. Prokaryotes use the same RNA polymerase to transcribe all of their genes. In E. coli, the polymerase is composed of 5 subunits, that is 2 alpha subunits, beta subunit, beta prime subunit and sigma subunit. Out of these 4 subunits, that is 2 alpha, beta subunit and beta prime subunit, that comprise the polymerase core enzyme. So, polymerase core enzyme that consists of only 4 subunits and sigma subunit is not included in polymerase core enzyme. These subunits, they assemble every time when the gene is transcribed, and they disassemble once the transcription is complete. Each subunit has a unique role. The 2 alpha subunits, they are necessary to assemble polymerase on the DNA. Beta subunit binds to the ribonucleoside triphosphate that will become part of the nascent newborn mRNA molecule, and beta prime subunit that binds the DNA template strand, and the 5th subunit, which is called sigma subunit, that is involved in transcription initiation. RNA polymerase enzyme that begins the synthesis of mRNA from an appropriate initiation site. Without sigma subunit, core enzyme would transcribe from random sites and would produce mRNA molecules that specified protein gibberish, nonsense protein. So, the polymerase comprised of all 5 subunits, that is known as hollow enzyme. What is a promoter? A promoter is a DNA sequence where transcription machinery binds and initiates the transcription. In most cases, promoters, they exist upstream of genes they regulate. The specific sequence of promoter is very important because it determines whether the corresponding gene is transcribed all the time, some of the time or infrequently. So, gene transcription is determined by the specific sequence of promoters. Promoters vary among prokaryotic genomes, but few elements are conserved like -10 and -35 regions upstream of initiation site. There are 2 promoters consensus sequences. -10 consensus sequence, that is called -10 region, that is TATAAT. And -35 consensus sequence, that is called -35 region, that is TTGACG. So these sequences in the promoters are recognized and bound by sigma subunit. Once this interaction is made between the sigma factor and -10 region or sigma factor and -35 region, then the core enzyme that binds to the site. Adenine thymine rich -10 region, it facilitates the unwinding of DNA template strand and several phosphodiester bonds are made, and the transcription initiation phase that ends with the production of abortive transcript, which are polymers of approximately 10 nucleotides that are made and released. So what is abortive transcription? Abortive transcription is also known as abortive initiation. It is an early process of genetic transcription. RNA polymerase synthesis and releases short mRNA molecule, and later on this RNA polymerase, it escapes the promoter region and enters into the elongation step of transcription. So this short mRNA molecule, which is synthesized and released from the promoter region, is called abortive transcription. To sum up, in prokaryotes, transcription, translation and mRNA degradation that takes place simultaneously. Polycistronic mRNA is found in prokaryotes, and eukaryotes they have monocistronic mRNA. So polycistronic mRNA that encodes for multiple protein products, and monocistronic mRNA that only encodes for one protein product. RNA polymerase is composed of 5 subunits, that is 2 alpha subunit, beta subunit, beta prime subunit and sigma subunit. Out of 5, 2 alpha, beta subunit and beta prime subunit, that is called core enzyme, and RNA polymerase with 5 subunits, that is called holoenzyme. A promoter is a DNA sequence where transcription machinery binds and initiates transcription. The nucleotide pair in the DNA double helix that corresponds to the site from which first 5' mRNA nucleotide is transcribed, that is called +1 site. It is also known as initiation site. And nucleotides preceding the initiation site are given negative numbers, and they are called upstream nucleotides. Nucleotides following the initiation site are given positive numbers, and they are called downstream nucleotides. So the name of these sequences like -10 and -35 refers to their position in the promoter region, that these sequences are located before the +1 site or initiation site. Abortive initiation is also known as abortive transcription. So RNA polymerase that binds to the promoter DNA to form RNA polymerase promoter closed complex. Then this RNA polymerase, it synthesis and releases a short mRNA molecule. This is called abortive transcription. And after the synthesis of this abortive transcription, this RNA polymerase depart the promoter and it moves to the next part, that is called elongation step of transcription.